From the Donald Danforth Plant Science Center, HEC-TV, St. Louis Public Radio, and the Danforth Center present Conversations, a discussion about people, plants, and partnerships. The Mizzou Connection.
I was going to remind you, I'm Jim Davis, just for the record.
Well, let me do a Sarah Palin thing here.
And
right?
But I would simply note that Ira Flato is used to being the moderator or the interviewer, and for the next hour he gets to experience some role reversal.
See it.
So
if I were to write another book, I kind of think about putting together all these myths that we take for granted as being true, that are really not true, and I think I could collect maybe a dozen of them. Start with the myth of why airplanes fly.
And the title would be What You Know That Isn't So.
Oh, write that down.
I kind of feel like, we've got this tortoise and hare effect, which is, he's the hare and I'm the tortoise, and the only way I'm going to keep up with him is if he lays down and takes a nap here on the stage, because...
I'm sitting here trying to think what my role is.
Yes. But
that one I'm going to turn over to you, Michael.
Well, touche.
This is a little bit like a tennis match, going back and forth. So the rate at which we're doing bad things is slowing down.
I would agree with that.
Yeah.
For a long time, I've often thought about my garden, my home garden, as a salad garden. Now, should I think of it possibly as a medicine chest garden?
It depends what you're growing in your garden, Jim, okay?
I just want to tell you a few more words about Jim Davis, for whom this stage was named and whom you've just seen. He was an academic, a political scientist, a teacher, a great university citizen. When there was an issue at Washington University, where I worked for many years, we called on Jim. If it were complex, if it were something new that nobody knew quite how to do, and he always responded. He was always there. If it was from leading the art school when we needed a new dean to fill in, running the undergraduate admissions office, starting the teaching center, writing editorials for a resurrected St. Louis newspaper. He built the first staff for my brother when he became a new senator in Washington. It was one of the best there was. He entered every new responsibility with good humor, enthusiasm, and hard work. First, to understand what was really going on, and then to do the job really well. But he never gave second place to his lovely wife, Jean, who's with us tonight. He never forgot that he was father, grandfather, and family man. I was amazed at the breadth of his knowledge, his ability to work hard, to master one area of study after another, his skill at transforming theory into practice, his understanding of the human response, and he could explain clearly and inspire trust, and as you've heard, do it with great humor. It became natural to call on Jim for help, and calling on him was so easy. He never complained. He was never too busy. He enjoyed new challenges, new opportunities to learn about new fields, to master new groups and to teach new groups. Challenges made his life more fun and more meaningful, just as he made the life more fun and meaningful for all of us. A great person, and I certainly miss him.
Good evening, and thank you, Dr. Danforth, for those wonderful remarks about Jim Davis. I've attended many of the conversations programs that he moderated, and he really was terrific. I'm Hal Burrows. I'm the chairman of the Conversations Committee here at the Danforth Center, and it's certainly a pleasure to welcome such a robust crowd here this evening. Tonight we're here to talk about the partnership between the University of Missouri and the Danforth Center, and how it is impacting plant science research and our region. Thank you to our Friends Committee, we had a meeting just before this in fact, and to the Conversations Committee for their continued support and commitment to this program, and to an ongoing ability to come up with some pretty interesting topics. The mission of the Donald Danforth Plant Science Center is to improve the human condition through plant science, feed the hungry, and improve human health, preserve and renew our environment, and enhance the St. Louis region as a world center for plant science. Those modest goals are the focus of this evening's conversation. And I would also like to thank this evening's sponsors, the St. Louis Public Radio and HEC-TV. Tonight's program is being recorded by the HEC, easy for me to say, and will event premiere on Sunday, September 11th at 5:00 p.m. It will continue to run on Sundays at 5 p.m. and Tuesdays at 8 a.m. the month of September. And past programs are available on the HEC website. Thanks to a supportive community, the Danforth Center can not only host these terrific programs, but can carry out its important work. And I do have to everybody's asked, is this the biggest crowd we've ever had? I think it's entirely possible that that's the case. If you're here for the first time, please raise your hand. I'll clap further away next time, sorry. That's terrific. Thank you all for being here. I promise you they got lots of pictures. They know who you are. If you're not already involved in the Danforth Center, there is information up front by the front door. There's a table there where there's more information. We do welcome newbies, as the case may be, and it's certainly terrific to see such a great turnout. Let's see. During tonight's program, this is, I know, Dr. Carrington's favorite part. During tonight's program, we will incorporate Twitter as a means to collect questions for our distinguished panel from the audience, as well as from others who will follow the conversation remotely. For those of you who tweet, please tweet us your question at Danforth Center and view selected tweets on our tweet wall. The hashtag for the event is hashtag CONVOSTL. For those of you who do not tweet, no worry. We will still collect question cards the old-fashioned way and transfer your questions to Twitter. Okay, instructions for how this will be accomplished are included on your question card. Do not ask me. The use of Twitter is a way to share the discussion and expand awareness of the exciting things happening at the Danforth Center and in our region. And I know somebody who posted a question here last time on Twitter, and he had to acknowledge that he couldn't post it on Twitter. He had submitted a handwritten question, and then it showed up on Twitter. I complimented him. I was so impressed. I would like to introduce at this time Michael Middleton, who's the interim president of the MU system. Thank you for being here this evening. When I moved here from Ann Arbor 26 years ago, I was thrilled at the Michigan fan base that St. Louis obviously had to offer. I eventually figured out that those hats were actually black and gold, not maize and blue. So, live and learn. But anyway, now truly the best part is to introduce the panel this evening. And first of all, Dudley McCarter, the past president of the Mizzou Alumni Association and tri-chair of Missouri 100, which is an organization a group that advises and assists the president of the University of Missouri in promoting the critical role of the University of Missouri system in our state's future and its reputation around the world. And and Dudley's also the founding member of the Bear, McCarter and Potter law firm. I got to know Dudley when he was the city attorney for the city of Creve Coeur back when we were doing some of the work for the Danforth Center, and that was certainly very entertaining. And our distinguished panelists are Hank Foley, the interim chancellor of the University of Missouri, and Blake Meyers, Ph.D., principal investigator and full member of the Donald Danforth Plant Science Center, and member professor, Division of Plant Sciences, University of Missouri. And without further ado.
Thank you, Hal. Let me add a footnote to what Hal said. He was the one who presented to the city of Creve Coeur the plans for this facility. And in the 15 years that I served as city attorney for Creve Coeur, no one made a more professional presentation than Hal Burrows. So. You were wrong about one thing, Hal. What you see here today is far more magnificent than you could have ever described that night to the Creve Coeur City Council, so I commend you on that. Right before I stepped on stage, my wife tapped me on the shoulder and she said, "Honey, I want to remind you of two things. Number one, you're no Bob Costas. And number two, not a single person in this audience came to hear you tonight." Good advice, honey. Thank you. I'm really thrilled to be here. This is really a dream come true for me, and I'm thrilled to introduce our two special guests. First, Hank Foley, interim chancellor of the University of Missouri. Hank graduated from Penn State, probably among other universities, but he received his Ph.D. from Penn State. He's a member of the National Academy of Inventors. He's a fellow of the American Association for the Advancement of Science. He has more awards than we could list tonight. He's the author of 120 scholarly articles. He holds 16 patents, and he's authored a college textbook on chemical engineering. Way to go, Hal, or Hank. I mean, Hank. Sorry.
Thank you.
And Blake Meyers came here from the University of Delaware. He received his BA from the University of Chicago, his master's and PhD from the University of California, Davis. He was chair of the Department of Plant and Soil Sciences at the University of Delaware. And his research focuses on genome-scale studies of RNA silencing pathways. Now, RNA looks to me a lot like DNA, except there's an R there instead of a D. And my guess is, before the evening's over, you're going to explain the difference.
Well, I don't know if we need to get into that, but I'd be happy to.
Let me start with Hank Foley.
Sure.
Hank, the University of Missouri at Columbia, it's a land-grant university, oldest university west of the Mississippi River, a member of the AAU, the American Association of Research Universities. Tell us what this partnership with the Danforth Center means to plant science research at the University of Missouri and plant science research across the country.
So this partnership started many years ago, but really started to blossom a few years ago when Jim came into the picture. And when we started to talk, I think 3 or so years ago, about really trying to bring the two institutions closer together. My motivation for doing that at the time before I was interim was the fact that I really wanted to see us do much, much better basic science at Mizzou so that we would in fact be safer with respect to the future of our membership in the Association of American Universities. And I was brought here really from Penn State to work on that issue and to be sure that we did everything we could to maintain our stature in that organization, which we've been a member of for 105 years. So I did a pretty quick analysis of where we were and what we were really good at, because I thought the resources that we had to put in those places where we'd get the best return on investment from an academic perspective. And it turned out that was easy to identify. The plant sciences and animal sciences were really right at the very top of the list of things that we do and we do well. We have 120 people who are involved in the plant sciences. We have 19 different experimental stations around the state where we take basic science out to the field. We sort of learn how to work with these new organisms in the field, and then they could go into agricultural production. So it seemed to me that this was logical. And at the same time, being a chemical engineer, I knew St. Louis as a city of science. And my familiarity with St. Louis goes back to Monsanto as the chemical company. So I'm not a plant scientist, but I think of these plants as little chemical plants, if you will, and that's really what they are. And I've tried to explain that to Blake. We haven't quite got there yet. We're not quite seeing it eye to eye, but that's all they really are, except you cut them down at the end, and you don't put quite as much capital in the ground. But to me, when I taught chemical engineering, and my field is catalysis, a catalyst is an agent that makes a chemical reaction take place that otherwise would not take place, that's not consumed in the reaction, but helps it to turn over many, many times. And catalysts are used throughout the chemical industry, the petroleum industry, and the pharmaceutical industry. But probably the premier brand of catalysis was done right here in St. Louis at Monsanto. And Knowles, who Dr. Danforth knew and others know that name, who was a National Academy member, was from Monsanto and actually worked on the first process, chemical catalytic process, interestingly enough, for the production of acetic acid from methanol. Absolutely shook the world from the 1960s onward and invented the whole science of organometallic chemistry, which I studied. What's interesting about that is acetic acid until that time had been produced biochemically, right? It had been produced by organisms. But the production rates weren't high enough, and the world needed more acetic acid for polymers. Monsanto engineered the first chemical process using a molecular catalyst that mimicked the kinds of catalysts you find in our bodies, in plants, and in the natural world. Absolutely astonishing. Well, fast forward, by the 1990s, being the kind of company that I guess Monsanto is, they realized that the future was in plant science. So you could have knocked most of us over with a feather duster when they decided to move from chemical sciences to plant sciences. So all of that was in my background, and I understood that. So when I came here, I saw Mizzou's strength in plant science, and I knew that DuPont was here, I knew that Monsanto was here, I knew that KSW was here, and we realized that St. Louis was becoming the epicenter of plant science. This institute, there is no other institute like this in the world. It's one of a kind, thanks to the people who are here and the generosity of this community to make it happen. And I just thought that there would be no better way for us to really improve and even make better the science that we already do in plants and around plants if we could affiliate. And thankfully, Jim was really very welcoming of that. My predecessor was interested in making that happen. He didn't do everything wrong. He got this right. And he made this happen to his credit. And the plan is very simple. The plan is to co-hire right now 4 different people. 2 will be stationed at Mizzou. 2, like Blake, will be stationed here. And then we're going to make it work between the 2 campuses. All right, so we are really committed to this. We're putting a lot of resources into it. And I think the future bodes well. I really believe that plant science is going to be the solution to world hunger, pollution, and probably material science as we go forward. And at the bottom of it all, if you look at how plants convert carbon dioxide, water, and sunlight into tissues and the like, it's all about catalysis. So I knew we couldn't go wrong. Is that good enough?
Would you mind if I ask Blake a question?
All right, I get
No. I sat at a football game with Hank, and
I'm sorry, I'm not good at short answers.
I knew that.
That's why I'm interim. There's only so much they can take.
Blake, you're at University of Delaware for 14 years, chair of the department. Your wife, your 12-year-old daughter, your dog, we're all happy there. Why did you choose to come here?
Well, I'll try and stay away from the chemistry side of things, but there was a really good chemistry, I think, between me and the opportunity here. I think Hank really covered the three major reasons why I came. University of Missouri, an incredible history in plant science that goes back a century or so. And just a rich history of discovery, agricultural research, impact, and tremendous faculty are there today, including a number of my collaborators. Of course, the Danforth Center, it's an amazing place to work. Infrastructure is remarkable. The colleagues that I have, the people that I can recruit to my group because of what's here, it's just a remarkable place to work. And then the region with the industry that Hank described, Monsanto, KWS, what you see in Bridge Park behind us, the Helix Center, Cortex downtown. There's really a strength in biotechnology and particularly plant biology in the area. It goes back decades. And so I think for me, being here, connecting with University of Missouri, being at the Danforth Center, having the opportunity to interact with industry scientists, the importance of agriculture and the recognition of the importance of agriculture in the region, plant science generally at the Missouri Botanical Gardens, and the appreciation for plants, it was really just an excellent fit for me. And so when I came here, when I saw what was being developed and saw this sort of synergy that was emerging between the two institutions and what's available in the region, it just felt like a natural fit for me. And so I moved, and here I am.
Well, we're thrilled you're here. And your family
Likewise.
adjusted to the cool summer that we've had this year?
Well, my family stayed behind in Delaware until the summer so that my daughter could finish 6th grade. And then my wife, being the very smart woman that she is, went on vacation with my daughter to Italy for 5 weeks. Meanwhile, I got the dog that you mentioned, the hamster, her car, and it turned out the air conditioning broke about 2 hours into Pennsylvania. And the 60 to 70 boxes that the movers brought in. And then I was able to take care of that, and then they arrived in July, right as the heat was at a maximum. But we've really liked it. We've joined a local pool, and schools are great. Started a little bit early for us. In Delaware, they always waited until after Labor Day. My wife is Italian, and August 15th is a national day of holiday on which nobody can do anything other than be on vacation. And so last year the schools here started, I think it was August 13th or 14th, and she looked at that and she said, oh, that won't work. Fortunately this year it was August 16th.
But be honest, I lived in Delaware for 14 years. I was in the Chemie department. It's another connection we have.
That's right.
The summers in Delaware are even worse, I think. They were worse than here. The Native Americans called the Delaware Valley the Valley of Sickness. Just so.
Another question for Hank, and if you could answer this even more briefly than the last question, we'll have an opportunity for
without
Blake.
mentioning acetic acid.
That's going to be hard.
Hank, I understand the University of Missouri has recently invested millions of dollars in the development of P.E.T. Center. Should I say that? P.E.T. CT?
Doesn't matter. That's
P.E.T. CT Center. Yeah. We've also recruited 2 radiochemists from the Brookhaven National Lab. First, tell the audience and me, because I don't know what it is, what's a PET-CAT Center? And
I have to be the straight man all night. Okay, I got it.
My dog and my hamster are really excited to hear the answer to this.
Yeah.
And Why is that going to be exciting for the
So-
Mizzou-Danforth partnership?
So by now you're thinking it's hundreds of little cages with places for your dogs and cats. P.E.T stands for positron emission tomography. And we decided that we would take some of the revenue that we've generated, and we've had good revenue in intellectual property over the last few years, and we would invest it back into ourselves. And no, this isn't going to be a short answer. I'm sorry.
You know what? I knew that.
Okay, so I'm going to try to shorten it up. First, let me point to Richard Ferrari. Richard, wave your hand. Richard came here from Brookhaven National Laboratories to set this up for us because we want to invest to make the world's best positron emission tomography, or PET, scanning system and center. And so we took money that didn't come from the state, but actually was generated off of our intellectual property in medicine, and we began to put it strategically into certain areas. My partner in this is Mark McIntosh, who's out of the medical school, and the genius who's making all of this happen is Dave Robertson, who's next to Richard Ferrari, because he kind of gets the big picture and he knows the people in the area. You may realize, and I'm trying to speak faster because he's looking at me, that we have a 10 megawatt reactor. And in fact, in that reactor, we produce all of the isotopes for North America that are used for imaging in medicine and are used for therapy in medicine. So every day, planes, jets are flying out of the Colubmia airport all over the country with various radioisotopes that are used in medicine. We're very proud of that. Because of that, we're also very good at taking radioisotopes and putting them into various kinds of compounds that are biologically active. Okay, put all these pieces together and we realized we could have a unique center. Plants, remember, take carbon dioxide from the air, water, and light and create tissues. What they really create are sugars. And one of the things you'd like to understand, as I understand it as a plant scientist and certainly as an engineer looking at plants, is what are the processes and what are the steps that take place in converting carbon dioxide, water, and light into plant tissue? What are the slow steps? What are the fast steps? How do the chemical reactions take place? But how do you get mass transfer to those reactions? How does transport and reaction take place in a plant? Well, the great thing about this is we can take carbon dioxide, and I told you it's not a quick answer.
I was afraid of that.
And we can label it with carbon-11. Carbon-11 is a radioisotope of carbon. You may remember some of that from high school chemistry if you haven't had chemistry in a long time. And it decays. And it decays to boron-11 because it's unstable. When it decays, it shoots out a positron. Well, we have technology that allows us to detect those positrons. Well, what that means then is we can do tomography, and we can actually look at where the carbon is and how it's radially distributed through, let's say, the stalk of the plant, through the leaf of the plant. But even better, we could take sugar molecules, and people like Dave know how to do the synthesis using carbon-11 radio isotopes, put those into molecules like sugars, and then inject them into the plant. The cool thing about this is we could do it with nitrogen-13 as well. It decays and forms positrons. Carbon, nitrogen, hydrogen, those are the building blocks of life. So suddenly we can watch these processes in real time. The half-life for carbon-11 is about 11 minutes, roughly 10, 11 minutes, 20 minutes. For nitrogen-13, it's about 10 minutes. So in fact, you have enough time to actually see transport through the plant, which is absolutely fantastic, absolutely fantastic. And Richard's experienced at this and has been doing it throughout his career, and now suddenly we've brought him here. We're marrying that to our other technologies, and we've got a source of funds that lets us push that forward. And I can tell you we're going to be the very best in the world at PET imaging. And we may push it to people as well, right? And animals, not just to plants, but first plants, don't worry.
My dog and hamster will be right in line, though.
Yeah.
So that's the Reader's Digest version of the PET Cat Center.
That's right. And I can't make it any simpler, but it is so exciting. And you saw some of it out here and some of the capacity and capability to do fundamental science that gives us. So it's not enough to just know about the chemical reactions, but it's important to know how the molecules get to the sites for those chemical reactions to take place.
I hadn't asked you another question, by the way.
Oh. Why, you know, I was trying to be clearer.
Thank you, thank you.
Blake, what's your degree in?
Not chemistry.
Don't say chemistry, yes.
Blake, since you got here, were your expectations met?
Expectations have been exceeded. Yes, it's.
And tell us how.
Well, the working here, as I mentioned, is really a remarkable place. I mean, the process of moving a laboratory is not easy. I have about 15 people in my group. Of those, 7 people moved with me, and so that was a process of packing up homes, packing up the laboratory, so freezers that needed to stay plugged in. So these are minus 80 Celsius, so they had to be on a truck with a power supply sufficient to run those freezers. We had a lot of important biological materials, USDA permits to go with that, to allow us to transport transgenic materials across state lines, bacteria. So it was a process of many months leading up to it. The actual act of moving, everybody getting in their cars, we sort of caravanned out here. The people in my group who didn't ask to move as the leader of the group. It was my decision, and so I pulled them along. And I think they've been thrilled to be here. I mean, immediately plugged into the center, plugged into the network of scientists. There's a steady stream of world-class speakers that come to visit here. Connections, of course, to University of Missouri. So people stop here, go out to Columbia, come back. So it's been really great for those types of collaborations. So then once we were here, the process of getting everything unpacked, buying new equipment, getting that up and going, getting back to the experiments, it went much more smoothly than I had anticipated.
Great. Hank, at the recent meeting of the Board of Curators, there was a presentation on a new $30 million improvement to our greenhouse facilities and plant growth facilities. Right. That has been moved up to the very top of the university's priority list. Tell us what that means for the future of the Mizzou-Danforth partnership.
It's just fantastic. So, you know, I want to point to Bob Sharp. He's our professor who runs the interdisciplinary plant science group and has been seminal in bringing all of this together many, many years before I ever got on the scene, and has done a lot of deep thinking about this, and it was really the person who identified Blake and brought him here. Blake, by the way, is probably one of the top 5 plant scientists under 45. Are you under 45 still? Or are you just over that now?
Let's say at 45.
Yeah. So when I met him, he was still under 45, and I was really excited about that. And so, but Bob is the one who sort of picks this talent and sees these people, and so I wanted to give him credit for that. But we realized, and Bob and his colleagues convinced me, that if we're going to continue to bring top-flight talent to Mizzou and into this partnership, then we've got to really be sure that we have the best growth chambers and greenhouses possible for these folks to do their experiments in. So that's a challenge for us, and we had to figure out how we could do this. And it's not easy. These are difficult times at public research universities. I don't probably need to tell you that. But we've come up with our own scheme, our own plan, if you will, for a way to facilitate the first phase of what would be a $90 million greenhouse project. And the first $30 million we'll draw from our own funds, from our own reserves, pending approval by the Board of Trustees at the October meeting. But they're very excited about it, so I'm quite certain that they will approve it and the President will approve it. And it's really interim. Just, you know, good in the science, good in the engineering, the politics not so good. But I'm really excited about that because that's going to make the difference for us. And so that will be how we make the next big step, because we want to go up the ladder, so to speak, with Danforth and be the premier university in the United States for plant science. We can't do that unless we have the infrastructure.
Yeah, I would add that continued investment in infrastructure, technologies, is incredibly important for the science that we do. I mean, we've become, as molecular biologists and my own work, very sort of reductionist. We're interested in small pieces of small components of organelles in the cells and understanding how they work. And so to do science at that level takes increasingly expensive, sensitive equipment and instrumentation. And at the same time, we have to marry that with things like greenhouses, growth chambers, where we have environments where we can grow the plants just in a very controlled way. And it's not cheap, but it's where we are in science. And so having those types of resources and equipment and facilities available to use allows us to sort of push to the edge of science, and it makes us more competitive for federal grants and allows us to do very exciting science, make discoveries in new areas.
Blake, I think you just touched on this, but let me follow up. What do you see for the future of this partnership? What collaborative projects do you anticipate building with the University of Missouri faculty? Yeah, well, I'd say part of what brought me here and attracted me to the position was this opportunity to work in a hybrid position of both academia at the university and here in a nonprofit sector at the Danforth Center. And so, on the university side, having that continued interaction with students and training them to go out in the workforce, to get those jobs at the cutting edge of science, using the infrastructure, the instrumentation that's out there, it's very exciting. And so I think of the opportunities in two areas. One of those is in training and education. How can we get graduate students from Columbia, where they're interacting with faculty and training there, how can we get them here to take advantage of the unique opportunities that the Danforth Center provides? And how can we then use that as a way to get them into industry positions that are found throughout the St. Louis area? So I think training is one part of that. And of course, research is another aspect of that. So in my work, I've got collaborations with a number of faculty members in Columbia. So there's a long and very successful program in maize or corn research, in soybean research. I've been working with Gary Stacy for a number of years, connecting his knowledge and background of the genetics and genomics of soybean with my interest in small RNAs, and that's led to a number of publications. So I'm really looking forward to, now that I'm here, it's easy to get in the car and drive over to Columbia, send my students there, my postdocs, and we can take advantage of the resources there and connect here. Great. I think a question came in from the World Wide Web that I'm supposed to read now. And, oh, it's the one up there. With Missouri leading in innovative technology and research to transform agriculture, what role can we fill in the next 10 to 20 years? Hank, you want to take a shot at it first?
Take a shot at it first. So again, I'm going to bring a historical perspective to this from the point of view of the chemical industry. In 1916... Catalysis and acetic acid again? You got it. You can't get away from it. We are right now at the beginning of a century of plant science that will lead to a plant science-based industry that I think is going to be very similar to the sort of progression we saw in the 20th century in the chemical industry, which became more and more sophisticated over time. I believe there will be plant science engineers just like there are chemical science engineers. And I think it's ironic and really interesting to realize that St. Louis was at the forefront of that revolution in the early 20th century, and they're at the forefront of this revolution in industry again at the beginning of the 21st century. What was the question again?
You could probably read it up there better than...
So that's my answer.
Wow. Wow, just like that, you're done?
I'm done.
Well, I would say, I mean, what you've described is essentially synthetic biology, which is the direction that we're going. And I think it's kind of a mantra, but the use of plants for food, fiber, fuel They are capturing energy from the sun and converting it to sugars, and I won't get into the level of detail that you did, Hank, but that's the bottom line, right? And so the number of uses, applications are quite diverse, and the companies that are in this region are working on all aspects of plant science.
So the biggest change in our lives, in my life, was watching PCR happen, right? Polymerase chain reaction changed everything. And then when I took biology, we still learned about the kingdoms, and we thought of them as separate. Today, I've learned that we could take genes from a human being, and we could splice that gene into a plant, and we could express proteins, right? Can't we?
Could do that.
We
with
could
the
do that in a
with
plant,
the appropriate
right?
permits.
And the like, right? With the appropriate permits. We aren't doing this. We're not doing this, but we could do this. But the point is that there's so much that could be done now that you can envision doing in pharmaceuticals, for example, that you never could have thought of. And we always had to think about a 20 or 30-step synthesis process that would take us from basic molecules to some complex molecule that would be a therapeutic. But today we're thinking about using plants, right, and expressing these things in plants and being able to create therapeutics that way. I think that's just astonishing and really exciting. And I really wish I were your age, not my age. You know?
45 is a great age.
It's a great age. I love 45. Yeah.
We have another question came in on Twitter. How will Mizzou choose what projects to do together with Danforth? Hank, you want to take a shot first?
Well, we'll let Mike choose. Mike Middleton, the
Middleton.
president. No, the way that's done, I think, is it's done by the scientists, right, Jim?
Mm-hmm.
I mean, the scientists make those decisions of what direction they've got to go. They've got to follow the science, and the science will take us to the right places.
Yeah, the decision is made through the hiring process, I would say, because as Hank mentions, the science is decided by essentially the interests of the individual. And so during the hiring process, you're looking for somebody that will bring something new to your institution, or institutions in this case. So how can you both strengthen what you have, but also bring some unique angle that will create synergies? And so I think that's what we're looking to create, hopefully through my hiring, but also through the additional positions that we'll fill in the coming years.
I think we have time for maybe one more, Jim, or we have one more. Another one just popped up. How does this research apply to practical use? Will it enable us to grow under harsh dry conditions? Blake, you want to take a shot at it first?
Sure. So I would say in my area, as I mentioned, a very, very reductionist work. But if we step back and ask, what are we aiming to do? So I study small RNAs, which are components of the cell that regulate gene expression, but I'm interested in the context of reproductive biology. So the basis of the hybrid corn industry is essentially reproductive biology. So Monsanto, Pioneer, they sell billions of dollars of hybrid seed. And so that requires taking two plants, taking the pollen from one, transferring it to the other. And so in my lab, we're studying how that pollen is produced and how we can maybe turn on the production of pollen or turn off the production of pollen so that we can refine the way that the plants are crossed, maybe do that in crops other than corn, in wheat, for example, and soybean, where hybrids are not used today. But as we need to feed and clothe and power a growing population, you know, we're looking at 9 billion people on the planet, ultimately a lot of that energy is going to have to come from plants, or a lot of those products. And so whatever we can do to boost the productivity of agriculture, I think is going to be quite important. And so that's where we're interested to apply the skills of my lab. With respect to harsh conditions, we also do some work on disease resistance. So a large fraction of plants that are grown die from pathogens, and in the developing world, it's a much greater proportion than in the developed countries where we can use chemicals or advanced germplasm. So we're studying aspects of that, and then I have a lot of wonderful colleagues here and in Columbia that are working on things like drought, heat, tolerance to adverse conditions.
So we have a major project, again led by Robert, John Walker, and others, which is NSF, National Science Foundation funded, on drought-resistant plants. Fantastic. And one of the great stories inside of that is we also have a 30-year relationship with the University of Western Cape in South Africa. And we are collaborating with the South Africans and trying to come up with new breeds of plants that, in fact, will withstand the kinds of changes we anticipate will happen in climates around the world over the next 100 years or so. But that's a wonderful story in and of itself. And one of the reasons that the greenhouse project got moved to the top of the list of projects that we're doing is several of our curators, one in particular, went with President Middleton to UWC this past summer, and while there, encountered the research that we're doing collaboratively with them on drought-resistant plants, and came back absolutely convinced that we had to move this forward, which helps this relationship as well.
Jim, are you, I don't want to eat into your time. This is, oh, well, let me ask one more then, if you don't mind. This will be for both Blake and Hank. What is unique about the Mizzou-Danforth partnership? What are the advantages to Danforth, and what are the advantages to the University of Missouri?
Let's start with the
I would say the whole relationship is unique, right? To have a university faculty member in a nonprofit where we're physically separated by, an hour and 45 minutes in the car, and to develop that relationship and in an environment in which there are these companies that we can interact with, the whole thing is unique. And that's really what drew me here and gets me excited every day.
It's very unique, but it is similar to one other program some of you may have heard of, and that's the Howard Hughes Medical Institute. And that was the model we had in mind. So if a faculty member is identified by the Howard Hughes Medical Institute as a top flight person that they want to support and be affiliated with, that's the kind of model that Jim and I had in mind when we put this together to try to bring people like Blake here, so that they would have both the opportunity to work at a university, but also the opportunity to be absolutely at the apex of research in plant science, just like the Howard Hughes is at the apex in medical science.
Great. Jim, I think the floor is yours, finally.
Please join me in thanking our panel. Actually, Dudley, I do have a few things to say. First of all, thank you. This is your first shot at moderating one of our conversations. Thank you so much for doing it. We've known you over 5 years now. We got to know you right after Terry and I moved here, and I can tell you and the audience you're a real gem here in St. Louis. Thank you for everything that you do. Blake Meyers, thank you for coming to the Danforth Center. Thank you for doing this tonight. Blake, I see Blake every day, at least every day he's in town, and it's a real treat to have you at the center. Hank, thank you so much for doing this. I do have a story. I don't want it seems like people have been picking on Hank, and I don't want to do that. I do want to tell a quick story though. And when we were recruiting Blake, we went to visit Hank in his office at University of Missouri, and Blake and I went in the office. Hey, Hank, how you doing? And within about 10 seconds, he said, wait here. He ran to the back of his office, reached his hand into a box, and pulled out 2 copies of a book that had a word in the title I can't repeat, but the gist of the book is how not to be a jerk. And he gave copies to me and Blake, and to this day, we can't figure out why you did that.
I was really sweating at that point. Here I am, a job candidate, and I'm getting this book thinking, oh, where's this going?
I can tell you for certain, if it wasn't for Hank, the arrangement and the partnership that we developed with the University of Missouri may not have happened, and if it would have happened, it wouldn't have happened as productively and quickly as it did. So thank you so much, Hank.
Thank you.
You know, it is all about partnerships. Why are we doing this?
Right?
It's about partnerships. Mizzou's been around since 1839. We've been around since 1998. This building wasn't constructed until 2001. Mizzou knows how to do a number of things, and plant science is one of those things. We partner with really great people like Bob and many of the other hundreds of scientists at Mizzou, and it's productive collaboration, and we can point to dozens and dozens and dozens of things that we do together. One of the questions was, how do you decide what to do together? How do you decide what to study? And the answer to that is pretty simple, and it's because we're both focused on using plant science to improve the human condition. That's actually our mission. Mizzou has a few other parts of their mission, but the plant scientists and the organizations at Mizzou share that vision. We tackle some of the most important problems that, if we can solve, agriculture, food security, the environment, and our industries here in Missouri are all going to be strengthened or more secure. That's really the guiding principle. How do we make water use more efficient to preserve our vital natural resources? How do we use things like genetics and more advanced breeding, like the type of technology that Blake studies, and that Mizzou is really one of the founding institutions. They're bedrock of genetic improvement of our major crop varieties. They invented much of it. When we get together and talk about what to do, it's focus on the big problems. That's the short answer. So, thanks again. Thank you all for coming, especially those who are here for the first time. Enjoy the rest of the evening and have a safe drive home. Thank you.